After studying this module, you shall be able to know about the methods of age estimation from teeth and difference between temporary and permanent teeth. The hardest and important part of the human oral cavity is teeth, which helps in mastication and also in speech or to provide structure to the face or oral cavity. Anatomically, tooth is divisible in two parts such as the crown and root. Clinically, crown is defined as the part which is invisible and fully covered by the enamel and actually referred to as the area above the cement enamel junction or the CEG of the tooth, whereas the portion of root is defined as the region which is below the CEG or the cemento-enamel junction and anchors the tooth into a bony socket acknowledged as an alveolus. Tooth is covered with three layers which are defined as the pulp, dentine and enamel. Enamel is the outermost layer of the crown which provides hard and non-porous cap over the dentine and it is made up of hydroxyapatite. The pulp is the important layer for forensic expert because it is the region which is composed of soft connective tissues which is present in the middle of the tooth and odontoblast or the stem cells from the dentine of the tooth at the edge of the pulp and helps the expert in individualization by the DNA fingerprinting technique. Dentine layer is mineralized layer of tissue and is basically that layer which covers the pulp. This layer of tooth is very porous which allows flowing of all the nutrients and minerals produced in the pulp to spread through the tooth. Estimating age or establishing identity plays a crucial part in forensic analysis. Identification here means the recognition of an individual based on certain physical characters which are thought to be unique to that particular individual. Identification of any living or dead person can be established by the data such as race and sex, age, stature, complexion and features, fingerprints, teeth, handwriting, speech and voice. In this module, we will focus on age estimation by teeth as a characteristic feature for establishing identity of living or dead. Age estimation is an important aspect of the forensic odontology. It is also used in the case of living persons when their age is disputed. While other methods are also in place to estimate the age of the individual, however, teeth are considered better in comparison to the bones. Age of the dentition is one of those aspects of physiological development that is uniform from the age of infancy till late adolescence. Now we will study about human dentition and the age estimation. The age of an individual up to 25 years can be determined within a range of 1 to 2 years from a combination of data provided by teeth, ossification of bone, height and weight, miscellaneous particular which includes birth record, changes occurring at puberty and changes due to old age. The information when considered individually is not of much value. For age estimation from teeth, it is necessary to know the difference between the two sets of teeth, the time of their eruption and the period when their root calcification is complete, and this can be ascertained on the X-ray examination as well. The two sets of teeth are temporary, deciduous or the milk teeth and the permanent teeth. Temporary teeth are 20 in number, that is 4 incisors, 2 canines, 4 molars in each jaws. Permanent teeth are 32 in number, that is 4 incisors, 2 canines or cuspids, 4 premolars or bicuspids and 6 molars in each jaw. Different types of methods such as the biochemical, radiographic, morphological and histological are used to estimate age from teeth. Age estimation from dentition can be categorized into three, that is, age estimation during prenatal, neonatal and the postnatal periods, estimation of age in children and adolescents, and the age estimation in adults. Now we will study about the age estimation during prenatal, neonatal and the postnatal periods. The primary tooth germ begins to form at 7th week of the intrauterine life or IU. The development of the enamel of all temporary teeth is generally complete by the end of the first year. At about 4 months of the intrauterine life, the germ formation for the first molar begins. In this group, the estimation of age could be pretty accurate. Histographic methods also help in the observation of tooth mineralization, that is 12 months prior to their appearance on the radiographs. The neonatal line is also thought to be as an indicator of birth. According to Bowerts, the slowdown in speed of growth rate of the enamel leads to the establishment of an apparent line of differentiation. Schiaparelli says that the neonatal could take up to 3 weeks after birth to fully appear. Therefore, the presence of the neonatal line does not indicate that the child was stillborn. However, the presence of neonatal line positively indicates a live birth. Sometimes, the forensic experts need to estimate the age from the skeletal remains also. In such cases, radiography and histological study is not effective. Alternatively, measuring the weight of the mineralized tooth cusp could help. At 6 months of the intrauterine life, a teeth weighs about 60 milligrams, 0.5 grams in a newborn baby and 1.8 grams at 6 months after the birth. Now, estimation of age in children and adolescents. Eruption and calcification of tooth are two instances which may be used to estimate the dental age in adolescents and children. Nystrom considers the eruption of teeth or tooth as a convenient option to estimate the age from dentition. It requires a greater degree of visual examination in comparison to expertise on the subject. The use of tooth eruption should only be limited to deciduous tooth because their eruption is genetically controlled. They start erupting at an age of 6 months and are completely erupted at an age of 2.5 years. The pattern of eruption of permanent tooth, on the other hand, is determined by on the various factors such as intraoral environment, infection, space and the premature loss of tooth. Therefore, the radiographic examination of the process of the calcification is a better alternative, as the calcification can be seen on radiographs for several years. It is not affected by factors like infection, space, etc. It also helps in the age estimation of the period during which no eruption takes place. Even an inexperienced odontologist could easily use and master this technique. Therefore, for the first 2 decades of life, the study of calcification is taken as one of the best indicators of age. Some of the other methods that are used for this purpose are, first is the Shocker and Maslow's method. They have explained 20 gradual stages of development of tooth starting from 4 months of intrauterine life till 20 years of age in the form of charts. These charts can be directly compared with the radiographs. Later, Ubelacker worked on these charts and included data related to the population studies. Differential development in males and females was also included, including the amount of variation. Bowers advises the use of modernized Ubelacker charts as the earliest form of the charts that had some drawbacks. Next is Demirjian's method. A notch-up system for the estimation of age was developed by Demirjian and his co-workers. the development of 7 teeth of the left mandibular was divided into 8 stages for each tooth named A to H. Here the third molars were excluded from the system, and later on Charlotte and Emergian, by further experimenting, decided to include them. A maturity score is assigned to each tooth which correlates to its developmental stage. A total for the entire 7 teeth is taken and plotted on an age conversion table. Keeping in mind the structural variation in males and females, the authors devised separate scores and tables for both the sexes. Next is use of third molars for the age estimation process. Though the third molar could be a good indicator for age between 16 to 23 years, after the development of the rest of the dentition, still its authenticity is always questionable because of greater variation in their morphology, position and age of formation. Based on the work of Kohler, Gunst and co-workers pointed out that by using a specific formulae, it can be said that whether an individual is of 18 years of age or not. After the calcification of all other 4 third molars, the probability of an individual being 18 years old is 96.3% for males and 95.1% for females. Most of such experiments have been carried out on the European populations, therefore it may vary when used in other regions. Now we will study about the age estimation in adults. Age estimation in adults is a difficult job as compared to younger age groups. Ritz and Associated have stated that after the completion of growth, the changes in dentition is affected by numerous internal and external factors such as nutrition, disease and the physical strain. Eruption of human dentition. The time of eruption of teeth gives a good indication of age up to 18 to 25 years, but there are variations depending upon the dietetic, geographic and other factors. In general, as compared to the permanent teeth, the temporary teeth are smaller except the molar, wider, more constricted at the neck, junction of the crown with the root and have jagged edges. The difference between the two sets of teeth and the approximate period of their eruption and root calcification has been shown in this table, where on one column we have the temporary teeth and on the other column we have shown the permanent teeth. So starting with the temporary teeth, it is small, narrow, light and delicate, which are longer than the permanent premolars, replacing them, whereas in case of permanent teeth it is big, broad, heavy and strong. In case of temporary teeth, crowns are china white in color. In case of permanent teeth, it is the crowns are ivory white in color. In case of temporary teeth, the junction of the crown with the fang often is marked by ridges, whereas in case of permanent teeth, it is the junction of the crown with the fangs are not so marked. In case of temporary teeth, neck are more constricted, whereas in case of permanent ones, the necks are less constricted. The edges are serrated in case of temporary teeth, whereas the edges are not serrated in case of permanent teeth. The temporary teeth has the anterior teeth vertical, whereas in case of the permanent teeth, the anterior teeth is usually inclined somewhat in forward direction. In temporary cases, the molars are usually larger, their crowns are flat and their roots are smaller and more divergent, while in permanent cases, the bicuspids which replace the temporary molars are usually smaller, their crowns have cusp which sharply differentiate them, their roots are bigger and relatively straight. Now, in this table, we have shown the temporary teeth, wherein when one column we have shown the temporary teeth, their eruption time on the next column and the complete root calcification age in the third column. Starting with the first, that is for the central incisors, the eruption time is 6 to 8 months, and for complete calcification 1.5 to 2 years. Next is central incisors. The eruption time is 7 to 9 months, and for complete root calcification time needed is 1/2 time to 2 years. For lateral incisors, eruption time needed is 7 to 9 months, and for complete root calcification it is 1.5 to 2 years. For lateral incisors, eruption time is 10 to 12 months, and for root calcification it is 1.5 to 2 years. For the first molars, eruption time needed is 12 to 14 months, and for root calcification it is 2 to 2.5 years. Next, for canines, eruption time is 17 to 18 months, and for root calcification the time needed is 2 to 2.5 years. For second molars, eruption time needed is 20 to 30 months, and the root calcification time is of 3 years. Generally, temporary dentition begins about the 6th month and is completed by the age of 2.5 years. The average child is expected to have 8 teeth at the age of 1 year, whereas 16 teeth expected at 1.5 year and 20 at 2 to 2.5 years. The temporary teeth begins to shed from the 6th to 7th year after the eruption of the first molar behind the second temporary molar tooth. The period of mixed dentition continues till the age of 12 to 13 years. Now students, in this table we have on one column the permanent teeth, the eruption time on the second column, and the complete root calcification on the third column. Starting with the first, it is the first molar. The eruption time needed is 6 to 7 years, and for root calcification it is 9 to 10 years. Then the central incisors, the eruption time is 6 to 8 years, and for root calcification it is 10 years. For lateral incisors, the eruption time needed is 8 to 9 years, and for root calcification it is 11 years. Then comes the first premolar. The eruption time needed is 9 to 11 years, and for root calcification it is 12 to 13 years. Next comes the second premolars. For eruption time 10 to 12 years, and for root calcification it is 13 to 14 years. Next are the canines. For eruption time needed, that is 11 to 12 years, and for root calcification it is 13 to 14 years. Next is the second molars. The eruption time needed is 12 to 14 years, and for root calcification the time required is 14 to 16 years. And lastly, the third molars. The eruption time is 17 to 21 years, and the root calcification time needed is 18 to 25 years. The permanent teeth appears first in lower jaw and then in upper jaw. The premolars are most irregular and are of little value in fixing the age. The first molar appears at the 6th year, and the eruption of the second molar takes place at about the age of 12th year. The eruption of the third molar or the wisdom tooth is very irregular. Due to evolutionary reduction in the length of the jawbone, the third molar may remain impacted, that is wedged behind the second molar and the jawbone. Therefore, the 12 years, that is the lengthening of the ramus of the lower jaw and behind the second molar, known as the space for the last molar, should be looked for. The existence of 4 wisdom teeth generally means that the subject has passed the age of 17 years. If the wisdom teeth are retained, an X-ray should be taken so as to ascertain if their roots are calcified or not. If they are not, there is a strong presumption that the age is below 25 years. Normally, the complete ossification of the root of the teeth takes place within 3 or 4 years of the date of eruption. The extent and degree of calcification can also thus help in the estimation of age. Next, we will study about the Gustafson's method. Gustafson devised a tremendous method of age estimation using 6 of these measures of the histological change. His method of estimating age consisted of microscopic examination of the longitudinal section of the central part of tooth in order to examine the changes in teeth as a result of wear and tear with advancing of age. Gustafson in the year 1950 developed this method for age estimation based on the histological and morphological changes of the teeth. He took into consideration the factors such as the amount of occlusal attrition denoted by capital A, coronal secondary dentine deposition denoted by capital S, loss of periodontal attachment denoted by capital P, cementum apposition at the root apex denoted by capital C, root reabsorption at the apex depicted by capital R, and the dentine translucency denoted by the capital T. Starting with the attrition that has been denoted by capital A. Attrition is the decreasing process of tooth structure by mechanical forces from opposing teeth. In this process, initially part of the tooth called enamel is affected and the level of attrition in enamel to downwards is pointed by Gustafson in the chart, that is A0 will denote no attrition, A1 will denote attrition limited to enamel level, A2 will denote attrition limited to dentine level, and A3 will denote attrition up to the pulp cavity. Next is the periodontal disease that has been denoted by capital P. It is a disease inflammatory in nature that affects the soft and hard structure that supports the teeth. Due to inflammation, the early stage is known as gingivitis. The gums become swollen and red, which is the body's natural response to the presence of harmful bacteria. And in this chart, it is denoted by P0, that is no obvious periodontal disease. P1 denotes the beginning of periodontal disease but no bone loss. P2 denotes the periodontal disease more than 1/3 of the root, and P3 denotes the periodontal disease more than 2/3 of the root. Next is the secondary dentine denoted by capital S. Here S0 denotes no secondary dentine formation. S1 denotes secondary dentine up to upper part of the pulp cavity. S2 denotes the secondary dentine up to 2/3 of the pulp cavity. And lastly, the S3 denotes the diffused calcification of entire pulp cavity. Next is root translucency, that is denoted by capital T. Transparency of the root means the ability of permitting light to pass through, diffusing it so that the object on the other side is not clearly visible. According to the root translucency chart, that is denoted by capital T, T0 denotes no translucency, T1 denotes the beginning of translucency, T2 is translucency more than 1/3 of the apical root, and T3 is the translucency more than 2/3 of the apical root. Next comes the cementum apposition denoted by capital C, wherein C0 denotes normal cementum, C1 denotes thickness of cementum more normal, C2 is the abnormal thickness of cementum near the apex of the root, and C3 is the generalized abnormal thickness of cementum throughout the apex of the root. Next comes the root resorption denoted by capital R. R0 here depicts the no resorption phase, R1 is spotted resorption, R2 is resorption limited to cementum, and R3 is resorption of the cementum and dentine both. The formula used is age is equal to 11.43 plus 4.56 into the total score, and such an examination is somewhat helpful to estimate the age between 25 to 60 years. Next, we will study about the dentine translucency. Bang and Ram were the first to use dentine translucency for the purpose of estimating the age and stated that root translucency is directly proportional to age, that is, it increases with age. Dentine starts to become translucent after 30 years of age from the apex. This alteration is because of the reduced diameter of dentinal tubules, which is caused by the increased intratubular calcification. The special formulae were devised to get a relative estimate of the age. Next is age estimation from incremental lines of cementum. Kagerer and group suggested that the age could be estimated from cellular cementum incremental lines. Authors claim that the accuracy within 2 to 3 years of the actual age. In addition to age, the incremental lines gave an indication of events like pregnancies, skeletal trauma and the renal disorders. Valenzuela and co-workers said that cementum apposition could also help in age estimation from the skeletal remains. This technique requires the extraction and sectioning of teeth, which is possible in the dead but not in the living individuals. Next is amino acid racemization. Human body uses amino acids exclusively in protein synthesis. Aspartic acid is also an amino acid which has a rapid rate of racemization. This rate is also high in dentine, therefore the teeth are a valuable source for age using this method. Next is the Boyd's method. Boyd has devised a method for accurate age estimation by means of the incremental lines. These lines can be seen on histological section of the teeth. They appear as cross striations on the enamel and represent the daily increments of growth. An exceptionally well marked line known as neonatal line is formed at birth. The number of cross striations from birth, that is the neonatal line, until the death is counted, and this provides the most accurate estimation of age. Now students, I will summarize all that we have studied in this particular module. The estimation of age is the most important aspect of forensic investigation in cases of mass disasters, where the personal identification is a crucial task for the expert. Age estimation from teeth can be done by studying the eruption sequence of the teeth in both phases of teeth, that is when teeth are temporary and when they become permanent. Age estimation up to 25 years can be learned through eruption sequence. The Gustafson is the person who devised a method for estimation of age. The method is based on the changes that occur in teeth with the age. The Gustafson's method is based on 6 changes, that is attrition or gradual wearing of teeth, periodontitis or loosening of teeth, secondary dentine formation or infilling of the normal root cavity, cementum apposition or increase in the tissue holding the root in place, root resorption and the root transparency. Boyd has formulated a method for exact estimation of age by means of the incremental lines. Teeth are an excellent source of both nuclear and mitochondrial DNA and have been successfully used in the forensic identification of compromised human skeletal remains. It is also clear that a complete understanding of the structure of teeth and composition along with the process of diagnosis in teeth is crucial for determining the location of DNA in post-mortem teeth cases.